overall goal of this video is to determine the application of serial section electron tomography to study the mitochondrial ultrastructure of Drosophila indirect flight muscle.
This method can help answer key questions in the field of structural cell biology, such as the structure and the functional relationship of mitochondria. The main advantage of this technique is that the cellular ultrastructure is revealed in 3 dimensions.
To begin the experiment, anesthetize 5 Drosophila specimens on ice and immerse each fly in 1 milliliter of 4% low-melting agarose in phosphate buffer. Allow the agarose to solidify on ice. Then, use a vibrating blade microtome to section agarose gel-embedded Drosophila into slices with 100 micrometer thickness. Immerse the slices in fixative solution containing 2.5% glutaraldehyde in 0.1 molar phosphate buffer. Wash the tissue sections in 3 drops of phosphate buffer, then follow with a wash with 2 drops of phosphate buffer with 20% BSA. Place the sections in gold carriers for high-pressure freezing, or HPF, filled with buffer and 20% BSA. Next, load the sample containing the carriers into a high-pressure freezer according to the user manual. After freezing, release the carriers from the holder under liquid nitrogen and transfer them to a freeze substitution device cooled to -140 degrees Celsius.